Okay, so to continue on with Chapter 17, specific defense of the host and the immune response. Antigen-antibody binding. We already talked about antigen and antibody, the relationship there. Antibody in our system, going to be attached to the B cell, going to be sitting, floating around in the circulation, looking for the foreign invader, the antigen, and then will attach to it, will attach to a specific portion of the antibody. And so let's take that to the next step and find out exactly what's going to happen to the antigen. Now, an antibody reacting to an antigen which will form a specific complex referred to as the antigen-antibody complex. Antigen-antibody binding is poor. The antibodies are said to have less of an affinity. Now, affinity is an attraction, and an attraction for the antibody toward the antigen. So there's an affinity there. So the antigen-antibody binding is poor. It is said to have less affinity. Protective effects of antibodies are due to several specific mechanisms, one of which is referred to as agglutination. And you may hear this term used as well in a clinical setting, agglutination. Agglutination, antibodies and antigens clump and are more easily ingested by the phagocytes. Another term, I think this is a test question, opsonization is basically when a bacterium, for example, is coated with an antibody that will enhance the destruction by phagocytic cells. So we see the bacterium actually becomes coated with the antibodies in our system. This tells the phagocytic cells to do their due, to come in, attack, and engulf and destroy the bacterium. In neutralization, antibodies inactivate viruses, toxins by attaching to them, and antibody-dependent cell-mediated cytotoxicity also allows the destruction of target cells too large for phagocytosis. So there are several mechanisms. Opsonization is just one of these mechanisms by which bacterium are recognized by the system and then allows phagocytosis to take place to be able to destroy these cells. Again, the antibodies will then attack and cover and enhance the ability of our cells to be able to carry this out. Antibodies coat the target cell, for example, a worm, and nonspecific immune cells release enzymes and other factors that cause its lysis. Antibodies that attach to the target cell in conjunction with the action of complements. Again, we're going to talk about complements in just a little bit. Complements are another part of our immune system that will help the immune system to be able to attack and destroy these foreign invaders. Now, inflammation, and I do want you to appreciate the process of inflammation. There is a couple of test questions about inflammation and what happens in inflammation. Caused by infection or tissue injury. Caused by infection or tissue injury. Every inflammatory process does not involve infection. Okay? Now, most infections involve an inflammatory process, but every type of inflammation, you twist your ankle and it swells, it's inflammation. No infection involved there. And so that's a common misunderstanding in the healthcare community that inflammation has to be related to infection. It does not. Leads in the inflammatory response, it leads to an attachment of complements to the microbe surface if it's a microbial infection or a microbial invader causing the inflammation. So, leads to the attachment of complements. Complements, in turn, lead to a process referred to as lysis, where it attracts phagocytes or other defensive immune system cells into the area. Their responsibility is to try to engulf, ingest, or destroy these microorganisms. This is a nice little table that's in your book that goes through these processes. You should know this table, know the terms that are related to this table. Agglutination enhances phagocytosis, reduces the number of infectious units to be dealt with, clumps them all together, gets them in the same proximity as the antibody so it can react on all of those substances together. There are laboratory studies that we do called agglutinations, cold agglutinations, so we can see if there's a clumping of cells together that are infected with different bacteria. Opsonization, we already talked about, coating of the antigen with particular antibodies that will enhance phagocytosis. Neutralization, which blocks the adhesion of bacteria and viruses to the mucosa. You know cranberry juice? Heard of cranberry juice? Why cranberry juice is important? Why is cranberry juice important? What? Say it one more time. Not cleanses your colon or liver. In urinary tract infections, I guess I'll be a little more specific. Cranberry juice has something in it called tannin. Tannin keeps the microorganisms from attaching to the mucosal walls in the urinary tract. So inside the bladder, in the ureters, that come from the kidneys, tannin in cranberry juice protects it. And that's not cran grape or cran orange or cran tangerine. I mean the generic cranberry juice, unsweetened. That's how it works. And so if you drink cranberry juice, it really does help. Also, you should be familiar with the activation of the complement system, which we're going to talk about in just a little bit. Cell lysis, the complements. Just remember, the complements are going to come in and help the immune system. Leads to eventually the lysis of bacterium. Then the inflammatory process, you should be familiar with disruption of the cells, basically by the complement reactive proteins, which will attract the phagocytic cells, the cells in our immune system, to come into the red blood cells, to get out into the tissue wherever the foreign invader is at, the antigen is at. Macrophages, different types of cells that we see in the differential or blood cell will come in and attack. And then the antibody-dependent cell-mediated cytotoxicity. Antibodies attach to the target cell, and then it causes a destruction by non-specific immune system cells to be able, again, to rid the system. So be familiar with the results of the antigen-antibody binding. Be familiar with each one of those steps and the importance of that. Laura, you had a question? Whatever your particular, the question was, is ocean spray cranberry juice good? But it's, as long as you get the unsweetened and it's pure cranberry juice, and they label theirs now. Ocean Spray does. They label theirs now and let you know. Yeah. All right. Yes.
As far as the inflammation process, do we have to know the different steps in inflammation?
One more time. In the formation process. Inflammation. Yeah, you should know the sequence of the steps. You should know what occurs first in the inflammatory response. Yeah, vasodilatation, for example, is early on in that process, so forth, so on. Okay? Now, immunological memory. Antibody titers is the amount of antibody present in the serum. The first contact with an antigen results in the primary response. A second exposure to an antigen results in an intensified response called secondary response, also known as memory or an anamnestic type of a response. Should be familiar with that term. Cell-mediated immunity, memory remains in a certain effector T cell that can distinguish for an extended period of time between host cells and foreign cells, always important. This is due to long-lived memory cells produced by a component of the primary response. Sorry. That's called blank. Now, in the system, in your human body, we are able to measure this memory. We refer to it as titer levels. Titer levels. So, if you receive a vaccination, for example, for hepatitis B, then several weeks later, several months later, I can draw serum from your blood and measure the level of antibodies that your system has produced, specifically for hepatitis B. And once you're at a certain level, a certain titer level, may vary amongst the types of organisms you get exposed to, but you should have a certain level. Once you reach that certain level, then you should have what we refer to as conferred immunity against certain types of diseases. And so we do that by measuring a titer level. The technique's been developed recently to allow the production of a large volume of antibodies in a test tube, which is an important factor in diagnostic study. In these techniques, for example, a cancerous cell and an antibody-secreting plasma cell can be taken from a mouse. Then you can immunize it and produce immunize it against a particular antigen. Now, again, this works in the test tube, and you'll see these types of hybrid types of studies that have been done where you actually can see whether or not certain types of cancer cells that are produced inside a laboratory animal, you can develop antibodies to that, and the antibodies will then attack these cancer cells. Now, obviously, this is important in doing cancer research. some of the problems related to this, and I'm not going to go into a lot of detail, and I didn't ask you any questions about it, but one of the problems associated with this types of technique, developing these hybrid models, is again certain disease processes that will occur in laboratory animals such as mice. You don't have that same mechanism in the human system, so you're not able to say, oh, well, it happened in a laboratory animal, so now we should be able to do that in a human as well. The systems are different. The immunological system of a mouse is different from that of a rat, which is different from that of a human. And so you can't always use these same models. You can get a lot of progressive information and excellent models done in laboratory animals, and that's what comes out on the media. Wow, they were able to cure lung cancer in a rat. Yeah, but you still can't do it in a human because the systems are different. The way our immune system has developed and the way the immune system of a rat or a mouse, if a mouse, because I thought they were pretty similar, a mouse and a rat, but they're not. Even their immune systems are different, and then take it another step to a higher level animal such as man, it's different as well. So many restrictions on using the gibbon ape now. The gibbon ape was really the best lab animal to do human studies on, but there are too many restrictions now, and they're very expensive to get the gibbon ape now, so we can't use that anymore. So anyway, but T-cell and cell-mediated immunity. So some forms of immunity can be transferred between animals only by transferring certain lymphocytes, hence the name cell-mediated immunity. Now what do we mean? When T cells form from the thymus, you remember that organ, stimulated by B antigens, it releases a certain protein. The proteins are referred to as cytokines. We know the best cytokine and that receives most of the attention in media is referred to as interleukins. Interleukins, the best known cytokine. I can't remember if that's a test question or not. It was certainly in the test bank. But when we talk about the best known cytokine, again, we're talking about chemically messenger immune cells. Best known cytokine is interleukins. Several representative interleukins function as follow. Cytokines are interferons, may be familiar with the term interferon, which helps protect us against viral infections. Interferons we'll come back to later. That may be a test question as to interferons, which help protect us against viral infections. We also see tumor necrosis factor, colony stimulating factor. These all stimulate the growth of infection-fighting cells that are in our system. So they're very important. Interferons were once thought to be the magic solution to forms of cancer and other types of infectious diseases. And interferons were used a lot, were synthetically manufactured and administered to individuals, and hopefully to be able to eradicate certain forms of cancer. But it really, the results never matched what it was once envisioned that they would be able to do. So cytokines, again, interleukins and interferons. Interferons help protect us against viral infections, but other cytokines, if I can ever say the word, cytokines and interferons are like tumor necrosis factor, colony stimulating factor, all of which help produce infection-fighting cells in our system. Additionally important cytokines include chemokines. They're from chemotaxis. They induce the migration of leukocytes to infected areas. You should know the term chemotaxis. I should have highlighted that for you. Chemotaxis. This is, sorry, I got a typo there too, but chemotaxis basically is the migration of leukocytes into an infected area. Again, these are the cells that are going to help fight off infections. Cytokines basically are soluble chemical messengers which cells of the immune system will communicate with each other, and again will help to mount that response, that inflammatory response that will come into various areas. Different types of interleukins. I didn't ask you any questions about this, but just for your own knowledge, interleukin 1, interleukin 2, 12, interferons. Interferons increase the activity of macrophages. You should be familiar with that. Chemokines cause leukocytes to move to infectious area. Just remember, chemokines means chemotaxis. Chemotaxis, leukocytes are moving into an area to fight off infection. T cells are the key component of cell-mediated responses. Developing from stem cells differentiate T cells from thymus cells. They migrate to lymph nodes, spleen, and other lymphoid organs. They enter into the lungs, gastrointestinal tract. They can use areas in the cells, particular types of folds referred to as Pyer patches. Pyer patches, those of you in A&P2 probably already familiar with Pyer patches in the gastrointestinal tract, but important in the lymphatic system. When stimulated by antigens, they differentiate into effector T cells to be able to mount an immune response. T cells have specificity for only single antigen. They don't recognize the receptors on the T cells. The T cells are unable to bind to the soluble antigens. Usually respond to only antigens processed by the specific system for that, which is the antigen-presenting system or the APC system, macrophages and dendritic cells. They must also have close association with the major histocompatibility complex antigens on the antigen-presenting cell. Now, this is very important when we are receiving organ systems from other individuals. Typically, if you're going to have an organ donated and or you're going to receive an organ, typically you go through what's referred to as major histocompatibility testing, MHC testing, to find out not only are you the same blood type, but do your antigen markers match up? Because as you can see, it's not now just as simple as having the same blood cells, but if your antigens are different from my antigen markers, then if I receive your blood, or I'm sorry, if I receive your organ, my immune system have antigen receptors, and it will circulate in our body, and if it recognizes antigen markers on your tissue that's different from mine, my antigen receptors are going to bind to it and launch an immune response and will reject the organ. So it's very important that you also go through major histocompatibility testing if you're going to receive an organ. The older you are, the higher the probability of organs not matching. The younger the recipient is, the better, because the young recipient has not had the opportunity to get exposed to a lot of antigens. So he's not going to have, that young individual, he or she, is not going to have as many antigen receptors developed because they haven't been exposed to a lot. Now, the older person has a much tougher time being able to receive organs from different people. Not that we don't do it, but it's much tougher because the number of exposures means that you have a higher number of antigen receptors, and so if you receive an organ, you may mount an immune response. And again, that's why a lot of individuals that receive organ transplants, they end up being on a number of medicines that suppress the immune system. And what we want to do is suppress our immune system. Corticosteroids, glucocorticoids are given to suppress our immune system so we won't mount an immune response to the organ that we have received. Cell-mediated immunity, specialized lymphocytes, mostly T cells, respond intracellular antigens. After differentiating in the thymus cell, the T cells then migrate to lymphoid tissue. T cells differentiate into effector T cells and then they are then stimulated by particular antigens. Some effector T cells become memory cells, and again, helps us to know what tissue is ours, what tissue is not ours. Also, again, pathogens that enter the gastrointestinal and respiratory tract can be affected by specific types of lymphoid tissue that help in that immune response. The Pyer patches were those structures that are located in the gastrointestinal tract in the cell. Nice little photograph of a dendritic cell presenting with an antigen. Moving right along. Helper T cells. Helper T cells I like to bring up because very important. You've already studied HIV, how it infects the cell, how it obtains an intracellular position and then an intranuclear position, and also affects our immune system. Primarily the helper T cells the helper T cells themselves help present T-dependent antigens to the B cells. So again, our systems are communicating. Remember, the cytokines are helping the immune system cells communicate. Helper T cells will then present these T-dependent antigens to the B cells, produce other helper T cells that respond to antigens. They also differentiate into two major populations. T helper cells, one, these cells activate cells related to cell-mediated immunity, such as macrophages, CD8 cells, we won't get into detail about that, T cells, and natural killer cells, all of which will attack these foreign antigens. T helper cell 2 type They produce cytokines. They cause B cells to begin producing eosinophils, which are protective against certain parasites. Eosinophils, IgM, and remember we talked about IgE, are important factors in allergenic reactions. Another point, and I can't remember again if that's on the test or not, but when we do the test review, you'll see eosinophils for parasitic infection, and I know we'll talk about it when we do digestive system, but the parasitic cells, the eosinophils will attack when we have parasitic infections. So helminth infections, intestinal worms, eosinophil count up, IgG up. You also see IgM is up as well, but IgE, eosinophils are the factors that we see that will respond in those types of infections. Cytotoxic T cells destroy target cells upon contact and will kill repeatedly if released. There are also important action against cancer, rejection of transplanted tissue, protection against intracellular bacterial and viral infections. Cytotoxic T cells recognize viral antigens on the host cell and cause destruction of that particular cell. And just as a review, helper T cells, two different types, cell-mediated immunity, type 2, eosinophils produced, eosinophils along with IgM, IgE, and then cytotoxic T cells destroy target cells, again by perfusing through porphyrin, which opens up the pores on those cells. You should be familiar with the concept of delayed hypersensitivity reaction. There are T cells, again, delayed hypersensitivity cells that are associated with allergic reactions such as poison ivy, and the rejection of transplanted tissue. These delayed hypersensitivity T cells or TD cells produce substances that recruit defensive cells such as macrophages. There are also suppressor T cells. The effect of them are not really well understood. We do know that they regulate the immune system or immune response when antigens are no longer present. Delayed hypersensitivity reactions, again, we see that in poison ivy, contact dermatitis, poison oak. Chemical contact sometimes cause these delayed type of hypersensitivity reactions. The tuberculin skin test is an example of that as well. You're given a little bit of an antigen, a little bit of a tuberculosis. This is a dead bacterium, just the coating cell of it. You give just a little bit, a little injection just under the skin. If you have been exposed to tuberculosis and you have antibodies in your system against tuberculosis, it will attack that little bit of antigen that's been injected under your skin and cause an inflammatory response. So you'll get a wheal, an indurated area of swelling just under the skin because your body already has antibodies to be able to attack that organism. Suppressor T cells turn off the immune system again, and antigen is no longer present. This is just complex of the system. You can review that. Again, I didn't ask you any questions from that effect. So nonspecific cellular components, the activation of macrophages. Now we know macrophages have the ability to carry out phagocytosis. The phagocytic capabilities of macrophages against intracellularly located viruses and bacteria and some cancer cells is increased when they ingest a target antigen and become an activated macrophage. So an activated macrophage has ingested a particular antigen, and now it's activated and will actually confer a degree of immunity throughout the system. Natural killer cells, certain lymphocytes called natural killer cells are capable of destroying other cells such as virus-infected cells, tumors that are not immunologically specific. They do not require any antigen stimulation. The contact and lyse target cells. Activated macrophages stimulate by ingesting antigen or by cytokines. And then natural killer cells, again, lymphocytes that destroy virus-infected cells and or even tumors. The production of antibodies, certain antigens, mainly proteins in combination with haptens, are associated with carrier proteins or carrier molecules or T-dependent antigens. In order for them to stimulate the formation of certain antibodies, they require the assistance of the helper T cells in the system. These helper T cells recognize complex antigens in that major histocompatibility complex, which means that cytokines, remember, are the chemicals that help these immune cells communicate with each other, such as interleukin-2 will activate certain B cells and become antigen-producing and or plasma cells in the system. There are T-independent antigens, just as a review, that are involved in this system as well. All of these types of T cells, again, are just a part of the immune system that will help destroy the invading antigens. An antibody-dependent cell-mediated cytotoxicity is especially useful in allowing our immune system to attach large organisms that typically our body can't form antibodies to destroy. That example would be the large parasitic worms. There are targeted cells that are coated with antibodies and oriented on specific regions of the antibody. The immune system cells known as natural killer cells or macrophages or neutrophils and are eosinophils, which we would see with parasitic infections. Receptors are a part of the FC region, or again a part of the antibody. And again, I didn't go into a lot of detail about the structure of the antibody, but basically will attach to the targeted cells, release substances that will basically lyse the targeted cells. Now, again, the body would produce a lot of IgE and would produce mast cells, but again, because parasitic infections are living organisms that also develop their own defenses, this type of a response may be able to wall off a parasitic infection, may be able to put a cell covering around it, but those organisms, they're not microorganisms. You can actually see them with your eyes, so they fall out of the context of microorganisms. But this antibody-dependent cell-mediated cytotoxicity may not be efficient enough to be able to completely destroy these